Good evening, everyone. I will, in this class lecture, describe the general characteristics, morphology, anatomy, and reproduction of Pinus. First, understand. First, the classifications of Pinus. First, we need to understand the classification of Pinus. Division, class, order, family, genus. Genus. Kingdom Plantae. Division Pinophyta. Class Pinopsida. Pinopsida. Order Pineals. Pineals. Family Pinacea. Genus Pinus. Some species are Pinus monophylla. It has single needle, and Pinus silvestris. It has two needle, and Pinus roxburghi. It has three needle. These are the species of Pinus plant. This is how it looks. The Pinus. These are leaves, foliage leaves, and also known as called as needles. It is of Pinus roxburghi because it has three needles. Pinus roxburghi. Scale leaves, long shoots, and these are dwarf shoots. General characters. Characteristics of Pinus plant. The genus Pinus is the most well-known representative of this family and has been known to mankind since time immemorial. Pinus is a beautiful plant. Pinus, a beautiful plant. Beautiful plant. Beautiful tall plant. It is a beautiful tall plant. Evergreen tree with horizontal branches arranged in holes, giving the tree a pyramidal appearance. In is represented by over 100 species. Morphology. Morphology root. The Pinus exhibits two types of roots. Long roots. Long roots having long roots having potential for indefinite growth constitute the main root system, while the lateral branches with restricted growth and relatively short life are the short root or dwarf roots. These are long roots and short Roots. Dwarf root. Stem. Stem of Pinus. The stem is erect, woody, and covered with rugged, scaly bark, which peels off. The branches are of two types. Branches are of two types. Long shoots, long shoots, and the dwarf shoots. Dwarf shoots. The long shoots or branches of unlimited growth bear an apical bud enclosed in bud scales. The dwarf shoots or branches of limited growth, also called short shoots. This is short shoots. It's long shoots. It's unlimited growth. Unlimited growth. Short shoots, limited growth. Also called as short shoots, brachyblasts or foliar spurs are borne on long shoots and arise in the axil of scale leaves. The leaf. The leaves on the dwarf shoots are of two types: 2 to 5 long needle like foliage leaves and the scale leaves of protective nature. The needle number per foliage leaf is constant for a species and is used as a taxonomic character. Example: Pinus monophylla, has one. Pinus silvestris has two leaves. It is only one needle or one leaf. Pinus silvestris has two or two leaves, two needles. Pinus roxburghi has three, and Pinus valiciana has five needles. The primary deciduous leaves, which are always single, appear soon after the germination and function as foliage leaves. The secondary permanent leaves on the needles are borne on short shoots or brachyblasts in bundle in the axils of scale leaves. In Pinus monophylla, the solitary needle is round. The needle is rounded. In the two needle pine, it is semicircular. In two needle, it is semicircular. In three needle pines, the Roxburghi, it is as triangular shape. This is a special characters of this unique Pinus needle. Anatomy. Anatomy of root. In Pinus, both long and short roots are dierds or tetrarch. The structure from periphery inward so that one layered epidermis is followed by multi-layered starch filled cortex having an outer zone of small and an inner zone of large parenchymatous cells. Mycorrhiza occurs in several conifers. In Pinus, it is well developed and has also been extensively studied. It exhibits well-developed ectotropic mycorrhiza association. Mycorrhizal association. Ectotrophic. The fungal hypha penetrate the intercellular spaces in the cortical cells of the root, forming the Hartig's net. The pine fungal relationship is symbiotic. Symbiotic. The pine tree benefits by the increased absorbing out of mycorrhizal roots and enhanced absorption of soil nutrients. Stem. Stem anatomy. In transverse section of a young stem of Pinus, we can see ridges and furrows formed due to outpressing of surrounding leaves. Epidermis is followed by a broad parenchymatous cortex, a ring of discrete collateral and open vascular bundles, and pith. It is pith. The stem is ridges and furrows. Pith and vascular bundle, open vascular bundles, and pith. Leaf. The epi and hypodermis layers are followed by mesophyll surrounding the diarch monarch stele. The mesophyll is chlorocymatous. Chlorocymatous with varying number of plate-like or peg-like. Plate-like or peg-like. Peg-like infoldings of the wall projecting into the cell cavity. Racing ducts are also present in the mesophyll. Racing ducts present in mesophyll. The endodermis is a single continuous layer of barrel-shaped cell. Endodermis, single continuing layer of barrel-shaped cell. Reproduction of Pinus. Pinus is monoecious. Is monoecious. Pinus is monoecious, but the male and female cones are borne on separate branches. The male cones arise on the lower and the female cones on the upper branches. Male cone and male gametophyte. The male cones which replace the dwarf shoot occur in clusters. Male cones, lower branches. Female cones, upper branches. They are monoecious, but the male and female cones born on separate branches. The males are on the lower and female cones are on the upper branches. The male cone and male gametophyte. The male cones which replace the dwarf shoot occur in clusters. Male gametophyte. The spore nucleus cut off a small lens-shaped prothelial cell toward the proximal end and large central cell on the distal end. Female cone and female gametophyte. Gametophyte. The female cone replaces the long shoots and the number of variable and may be up to 6. Each female cone consists of a central axis on which 80 to 90 megasporophyll ovuliferous scales or ovule-bearing scales are arranged in a spiral fashion. The bract and the ovuliferous scales together form a seed scale complex, megasporangium and micro megasporangiensis. Megasporangium and megasporangiensis. The ovules of Pinus are unitegmic and crassulate. Ovules unitegmic and crassinucellate. The integument is free from nucleus for most part of its length, except at the chahazal end. It forms a fairly broad micropylar tube which is curved in during pre-pollination stages but is curved out at the time of pollination. Female gametophyte. The functional megaspore enlarges considerably its nucleus. Female gametophyte, its nucleus divides mitotically to give rise to the free nucleus gametophyte. The number of nuclei in the free nuclear gametophytes is constant for a particular species. Pollination and fertilization. Pollination is wind. Pollination is wind pollinated. Wind pollinated. The yellow pollen is produced in such great abundance that is in a pine forest. It is called sulfur sour. Sulfur sour. At the this time, female cone is still open and the wrinkled pollen grains are easily sieved in, roll down the obliquely tilted scales, and finally come to rest in the of the micropyle vicinity of the micropyle. Vicinity of the micropyle. At this stage, the ovule secretes a drop of sugary liquid, the pollination drop. Pollination drop, which fills the micropylar cavity and exits out at the flared tips of the integument. Fertilization. The pollen tube enters the archegonium, forcing itself between neck cells. The archegonium. Pollen tube enters the archegonium itself between neck cells. The pollen tube, on bursting, releases two unequal male gametes, stalk cell and the tube nucleus with associated cytoplasm. The male nucleus enters the archegonium and moves towards egg nucleus and eventually fuses with it, forming the zygote. Fusion of male and female gametes syngamy, which results in the formation of zygote, is known as fertilization. The zygote represents the first cell of the sporophyte of next generation. This is the end process of fertilization. We have discussed the pollinations, mygasporium, reproduction is monoecious, anatomies, leaf structure, stem, general characteristics, long shoots, short shoots, the division, the studies. This is microsporophyll, pollen sacs. This is study of the Pinus of longitudinal structure of female cone. And this is the structure third year female cone. Mature female cone. Cone ovules. Yeah, backed scales. This is known as backed scales. Here, ovuloferous. This is known as oviparous scales. This is known as ovule. This is cone axis. The transverse section of dwarf shoots. The section almost resembles with that of the main stem. The outline is wavy due to encircling scaly leaves. The dwarf shoot. TS of dwarf shoot. Transverse section. The outline is wavy due to encircling scaly leaves. The tissue are differentiated into epidermis, cortex, and stele. Epidermis is made of single layer of thick walled cells. Cortex follows epidermis. Outer few layers close to epidermis are thick walled, whereas the inner layers are thin walled and parenchymatous. Differentiate into epidermis, cortex, and stele. About 6 resin canals are present in the cortex. Tannin cells are also irregularly scattered in this region. Stele is an ectophoic siphonostele. This is ectophoic siphonostele. Endodermis is single layered and is followed by pericycle. Both the layers are undistinguishable. Vascular bundles vary in number. They are generally 6. Each vascular bundle is conjoined, collateral, and dark and open. Male cone microsporophylls and megasporangium structure. Male cones replace the dwarf shoots. Each male cones arise in the axil of a scale leaf. The main shoot on which these are produced continues to grow further. Male cones are grouped in clusters on the shoots of the same year only. Each male cone has single centrally located cone axis around which scaly microsporophylls are spirally arranged. Each microsporophyll has an expanded triangular central part and stalk-like base. Terminal part projects into a tip. Few lowermost sporophylls are sterile. Few lowermost sporophylls, sterile, and do not bear any male reproductive structures. On the abaxial side, each microsporophyll bears two ovoid microsporangia or pollen sacs on its lateral sides. Each microsporangium has its own wall, which encloses many microspores. The young microspore is globular, spherical in shape and uninucleate. Longitudinal structure. This is female cone. Longitudinal structure of male cone. It shows a cone axis bearing microsporophyll. The cone axis is centrally located. Microsporophylls are spirally arranged. These are scaly, triangular, and expanded. It is attached to the cone axis by a stalk-like base. The outer expanded part is sterile and is known as apophysis. Microsporangia are present on the lower or abaxial surface. Each microsporangium has a wall that encloses a cavity. The wall consists of epidermis, wall layers, and tapetum. The cavity shows numerous microspores in various stages of development. Longitudinal structure of female cone. Of female cone. Female cone is made of centrally located cone axis. The centrally located cone axis, you can see here, and spirally arranged sporophylls. Each sporophyll consists of two kinds of paired scales, backed scale and cone scale. This is known as backed scales. This smaller is known as backed scale or also known as cone scale. Backed or cone scale. And this is known as ovuliferous ferrous scale or seminiferous scale. Many small and thin backed cells are arranged spirally around the cone axis. These are directly borne on the cone axis. Each of these is present on the abaxial lower side of the ovuliferous scale. On the abaxial upper side of the backed scale, a thick, large, woody triangular ovuliferous scale is present. The ovuliferous scale in the middle part of the cone are the largest and get gradually smaller towards its base and apex. Ovuliferous scale and backed scale are fused for a little distance near the cone axis will free at a distance away from it. Morphology of female cone. The female cones are larger. This female cone are larger than the male cones. They are born at the apices of the young elongated shoots, replacing the shoot of unlimited growth long shoot. Single shoot may bear 1 to 4 female cones, which are reddish green in color and mature in 3 years. Sir, reddish green in color, green in color and mature in 3 years. It's third year. Mature in third year. Pinus. Female cone. This is third year finest female cone. In the first year, cones are compact and sporophyll are closely arranged. The second year, the cones are large in size and woody in nature. Woody in nature, but sporophylls are still compactly arranged. In the third year, cone become loose. Sporophylls separate from one another due to elongation of the cone axis. In six, six one, each female cone. Cone consists of many sporophylls arranged spirally around the cone axis. This was discussed longitudinal structure of female cone. This is microsporophyll pollen sac. This is natural habitat. The three needle is known as Pinus roxburghi. It helps to identify the species. Thank you, everyone. In next class, I will explain the nitum, Gymnosperm nitum, its morphological characters, classification, anatomy, structure, reproduction, and the fertilization of pollinations. Meet you in the next class. Thank you. Like, share, subscribe.